What is your name? We've met a couple of times. Yeah? Ah, yes. Thank you. Un placer.
Tobias. You're Tobias?
Exacto.
Eso.
Exacto. Buenas. Angelito.
Ahorita parece alguien más.
Todavía yo lo voy a presentar en español y usted se presenta en inglés.
¿En inglés? Sí.
No, por eso, yo lo voy a presentar en español y
Ah,
usted
ok.
se presenta en inglés.
Sí.
Para que sea una dinámica agradable y yo no tenga que hablar inglés. Hay refrigerio si quieren, de atrás, que pueden tomar antes de la charla y hay café.
Eso está buenísimo. Refrigerio. Refrigerio de la tarde.
Sí, lo que pasa es que las empanadas recién hechas son crunch.
Hola.
Ya después de los platos.
¿Bien? ¿Usted? Bien.
Ya voy a agarrar una.
Todavía hay tiempo.
2 minutos.
Faltan 2 minutos.
Tranquilo.
La fresca, ¿no?
Cafecito de la tarde. Empanada de la tarde.
Sí.
¿Pueden tomar algo mientras inicie la presentación de ella? Vamos a ver qué. Vamos a ver.
Creo que empezar a las 3. Ya todavía no es la hora. No, es Tobías. Tobías fue investigador afiliado del Instituto Monteverde. 21, 22, ¿verdad? El 21.
21. Sí.
Y hizo su investigación, bueno, hizo varias cosas. Hoy nos va a hablar sobre eso. Más si lo quiere entrevistar, le dice. Cuando me canse de los salamos, porque no tengo un rato más aquí. No estamos
¿Qué tal?
No sé si empezamos, yo soy como histérica con eso. Esa es la 3 y 1. Bueno, buenas tardes, bienvenidos. Para los que no me conocen, mi nombre es Luisa Moreno, yo trabajo aquí soy coordinadora de investigación. Y para mí es un placer introducir a Tobías. Tobías fue investigador afiliado del Instituto Monteverde en el 2021-2022 para realizar sus estudios desde la Universidad de Copenhague y hizo diferentes estudios y hoy nos va a compartir sobre este tema. Él estaba estudiando para obtener su maestría en ecología y hoy me comentó que él se considera un ecólogo molecular.
Exacto. All right, so let's kick it off. Like she said so beautifully, I am from Denmark, and I've done this study, which I've called One Fungus to Rule the Frogs. And it is an eDNA assessment of the amphibian chytrid dynamics here in Monteverde and across several gradients such as elevation and canopy cover. Right, so let's kick into it. So first off, what is Batrachochytrium and dendrobatitis and the amphibian decline? Batrachochytrium dendrobatitis, which I will call BD from now on, is a chytrid fungus that is parasitizing on amphibians. It's infecting the skin of... Bienvenidos. Hola. Right. So, Batrachochytrium dendrobatitis, a fungus parasitizing on amphibians, is causing a lethal skin disease called chytridiomycosis, where the fungus infects the skin of the amphibians and disrupts homeostasis. So basically the controlling of the water within the frogs. Yes, it has a worldwide distribution since the 80s at least, where it has caused a severe amphibian decline. Globally, more than 90 species of amphibians, mainly frogs, have gone extinct, and at least 500 species are in severe decline. Here in Monteverde is one of the first places that it occurred, and it has caused initially a 40% decline in amphibian richness. And that's based on an amphibian survey during the 90s over a period of 5 years. And one of the species lost is, of course, the golden toad, which I assume many of you know about, along with others. Right. So a bit more about this fungus, which is a quite special one, I'd say. The group is called Chytridiomycota, coming from the Greek word chytridion, which translates into a little pot, because it creates this, you could call it a little pot, which spews out zoospores, that are these single cell spores with a little tail. It's very similar to a sperm cell in reality, and it can swim around in aquatic habitats and look for frogs to eat. There's 900 species described in the world, and there's definitely many, many more. People just haven't looked for them, and you can't see them because they're single-celled. Most of them are parasitizing on algae or oomycetes, which is also a fungus, or small invertebrates. The group Chytridiomycota is ancient. It's one of the first fungus groups. The first records are about 500 million years old, from the Precambrian era. BD, on the other hand, is a relatively new species. It's being debated when it occurred first, but it's not really known. Also, the exact origin of where it comes from is not known. A lot of people suggest Eastern Asia, but there's also evidence that it's from Southern Africa, so really no one knows exactly. It's asexual. It doesn't have sex. It simply just clones itself. And just to go through the life cycle, It starts out as this single-celled zoospore, which swims towards amphibians and finds a good place on the skin where it creates rhizoids, which is kind of like roots to make sure that it stays there. It then starts to clone itself, and after a couple of days, it will start spewing out new spores into the environment. And it is, for some frogs, highly lethal. In this picture, it says 120 days for Atelopus frog to die. I've read papers showing that in 40 days, it has a 100% mortality rate in other species. So it's pretty aggressive. Meanwhile, other species are not affected by it. They have tremendous amounts of the fungus living on them, but they don't get sick. Right. Something about serving BD and amphibians. As you probably know, there's a biodiversity crisis going on. And in order to combat the biodiversity crisis, we need to efficiently monitor biodiversity and the things that affect it. The issue with amphibians is that they're super hard to find. Imagine having to find a Vietnamese mossy frog on the bottom of a rainforest during night. You have to look really carefully. And similar to this one that looks very much like a leaf, other amphibians, they spend their entire life cycle in the canopy, and people don't even know that they're there. And also, they live in swampy areas that are hardly accessible. And in order to assess BD on the frogs, traditionally, you would need to catch a frog and take a Q-tip swab from it to see if it's infected by BD. So all in all, it's really hard to assess the distribution of the fungus and the amphibians as well, especially in a tropical setting. And that's where eDNA is a solution. Because you'd only need to take an environmental sample, regardless of frogs being present or not right of the moment that you're there, and you can see what's going on. So the aim of our study was to explore the use of eDNA to assess the presence of BD across various habitats and considering elevation and canopy cover. And we would use the same environmental samples to look at amphibian diversity and examine how BD impacts this diversity. Based on the literature, we expected that BD positive sites would have a lower amphibian diversity. This is not very surprising as it's an amphibian killing fungus. We expected that habitat differences would somehow affect the presence of the fungus, with the aquatic habitats having the most fungus, as chytrids are all mostly aquatic fungus. And we also expected that both altitude and canopy cover would increase the prevalence and infection load of BD across all habitats. And by infection load, I mean the abundance of the chytrid present in a given environment. Right, so a bit about eDNA and how we used it. Environmental DNA is simply defined as genetic material that is in the environment left by organisms. So whenever you go somewhere, you will lose some skin cells, some hair, whatever leftovers, mucus into the environment, and it is possible to detect those small fragments of DNA. We used qPCR, which is a technique specifically used to see how much of a given organism is present in an environment. So this technique is only focusing on one organism, in our case, BD. We also used metabarcoding and Illumina sequencing to assess amphibian diversity. Generally, eDNA has not been worked a lot with in the tropics. As it's a relatively new field, it is slowly moving here, but it's also a bit difficult to work with eDNA in the tropics because the climate is warm and it rains a lot, very often at least. And those factors contribute to a fast degeneration of DNA molecules in the environment. But still, it has been shown that if an animal of a relatively large size, like a mouse or whatever, have been in a body of water, you can detect its presence 5 to 7 days after it's been there. So it's still pretty good. I found this one on a website, and I thought it was pretty good at showing the workflow of working with eDNA. So to start here, we have this beautiful little pond where you take a sample. For aquatic samples, you will collect a lot of water and push it through a small filter which collects all the DNA fragments in that water. And that's really your sample. That's the filter. You then extract the DNA and you use certain primers to amplify the DNA you're looking for. A primer is a tool that helps you to detect a specific kind of organism. So in my case, I had 2 primers, one specific primer for BD, which only reacted to BD, and another, which is, you'd call it a universal primer, which only connects to things that are amphibians, but doesn't discriminate different amphibian species. But if you're not an amphibian, it will not connect with it. And then comes a very long, tiresome process of bioinformatics. And at the end, you see, whoopsie, there was a blue whale in this pond, which is not a very good representation, but that's how it works. So sampling. We had 7 locations, 3 Pacific, Reserva Aguti, Cerro Amigos, y Curicancha, and 3 on the Caribbean slope, San Gerardo, Poco Sol, y Finquestella. And 1 which I called southern, it's in Laguna Pecon, which is in Bosque Etanos Los Niños. And it is right on the rift, on this continental divide. So it was samples both from the Pacific slope and the Caribbean slope. But I would say the climate there mostly represented a Caribbean climate. Our habitats were ponds, streams, bromeliads, and in close proximity of each sample, we would also take a soil sample and a leaf litter sample to see what's going on there. And here's just some photos of what the different locations look like to give you an idea. This is Laguna Pecon, very beautiful, hard to reach. This one you may know, it's in Reserva Aguti. The streams varied also a lot, with some being like large and open, and others were slow with a closed canopy over them. And these are all things that you need to take into consideration when you look at your samples. The bromeliads, you may not know, but they are exclusively in the Americas. And they're this incredible habitat that collects small pools of water, and a lot of different animals like to live in those pools of water. Most of our bromeliads were terrestrially dwelling on the soil, but we also had a couple in the trees that were easy to reach. So, on to some results. So first of all, how did BD look across the different microhabitats? So here on the y-axis, we have the prevalence. So how many % of the samples were positive? And on the x-axis is the different is the bromeliads and the leaf litter samples. And the other group, high prevalence, is ponds and streams. So ant soil, which was a big surprise for us. We're not sure why we found this. there's a lot of relatives to BD living in the soil. So maybe our primers weren't specific enough to only attach to BD, but also attach to some soil-dwelling chytrids. Another reason could be that the soil is actually a really good container for DNA fragments. As there's not a lot of oxygen there, it's a stable climate. We don't know. It's weird that every single one of our samples were positive. Right. Next, so this is looking at the BD prevalence across our two continuous variables, canopy cover and elevation. And here we expected to find that as both of these increases, we will see an increase in prevalence. And that was truly the case for canopy cover. It's a nice steep slope. On the y-axis, we have the predicted probability of BD being present. So it clearly shows that as there's a closer canopy over a habitat, the probability of it being infected by BD increases. On contrary, we found the opposite with elevation. As elevation increased, the probability of BD being present decreased. And that is pretty weird, to be honest. We don't know why, because the high altitude sites, in theory, they should be really, really good habitat for BD. And previous studies have shown that especially the high altitude frogs are the ones that are suffering the worst from BD. So this is quite weird. In terms of canopy cover, we were expecting it because the closed canopy, it gives moist, cool conditions. That's something that the fungus really likes. Actually, some frogs, they seek out sunny locations when they sense that they're sick. They find a spot in the sun and heat up. We call it behavioral fever. And of course, that is harder to do if you're in a place with absolutely closed canopy. Right. So looking a bit more at BD and canopy cover, but this time at the infection load. So for each of the positive sample, how much of the fungus was present in that sample. And it shows the same pattern as we expected, that as canopy increases, the infection load also increases. And especially in leaf litter and bromeliads, they are severely affected. And if you think about the nature of those habitats, they're really small environments, and you could say they're drought prone. If you have sun shining on a patch of leaf litter or a small bromeliad, it's going to dry out pretty fast. And in contrast, soil and streams and ponds, they're much larger bodies, and it's going to require a longer time to heat them up and to dry them out to create a bad environment for BD. Secondly, it probably also is something to do with a dilution factor. So imagine a frog jumps into a bromeliad tank. It holds maybe 60 milliliters of water, and it's infected with BD. There's going to be a lot of fungus cells per water in that bromeliad tank. In contrary, if it jumps into a lake and the fungus disperses into the full body of the lake, the concentration is going to be super low, even though there's a lot of fungus living on that specific frog. But yes, the results are pretty clear that increasing canopy does indeed increase the abundance of BD. Right, on to some of the frogs. and the metabarcoding, the technique we use to assess amphibian diversity. Overall, we found 24 genera and 21 different species. And the reason that not all species were able to be determined to species level, but only determined to general level, has something to do with the primers that we used. We chose primers that were not very specific for amphibians. So this primer gave a larger probability of detecting that some amphibian is there, but it was worse at telling us specifically what species of amphibian was there. Metabarcoding was great at detecting difficult species, you would say. And by this, I mean either, in this case, Ismohyla tica, which is an endangered species. Monteverde region is one of the last refugees of this species. And it's a species that you don't see it every time you go out, far from. And it's a proof, it's a testament to the efficiency of eDNA, because I spent 1 minute in that location just taking a water sample, whereas as normal field service, you would spend a long time and you may not even find it. Another good example of the efficiency is the Hippopagus variolosus, a sheep frog, I think is the common name of it. They spend almost all their life cycle buried underground, and they come out to breed, and they breed in these small ephemeral, so temporary pools of water, and that's exactly where we detected it. And I talked with some of the frog guys here in the area, and they told me, this is a frog, it's really hard to find. We know it's here, and it's quite common, but you just don't see it very often. We also found a tendency that maybe some of the amphibians have adapted to live with the chytrid. And this is the case for Litobates, which is leopard frogs, I think. And they were present. It was by far the most common species in our study, and it was found throughout all the BD positive sites, while in some of the BD negative sites, it wasn't present. Right, some more about amphibian diversity. So the way these graphs work is that on the x-axis, we have the number of sampling units, with a sampling unit being an environmental sample. So either one filter from water or one soil sample or whatever. And here, the genus diversity is the amount of genuses found in that group. And we have two groups. The orange line represents all the BD negative sites, and the blue line represents the BD positive sites. So starting with the small graph here, this is created using only what you call richness. So richness is the total number of species found. You don't put into account the abundance of each species, which is something you're also interested in if you want to assess the biodiversity of a given community, right? And looking at this, there's no clear difference. And that aligns well with the literature, actually. I've been told that the amphibian diversity here in Monteverde now is more or less back to the same as it was before the initial decline. But the issue is that it's generalist species. So we've lost the rare species and new species have come in, and these are generalist common species. And you could say they're not as interesting or unique to the area as the ones we've lost, right? This graph here, the lines are based on what you call Shannon diversity. And Shannon diversity is an index for measuring biodiversity. And what it does is that it takes into the account the abundance or frequency that a species occur. And here we see a pretty clear pattern that the BD negative sites have a higher diversity than the BD positive sites. And this is probably part due to Lithobatus, the frog I was telling about previous slide, that certain frogs, amphibians, are becoming much more normal. So even though we have the same number of species, the community is changing when the fungus is present. And instead of having a diverse community with more or less equal amounts of each species, there are the same number of species, but a few species are dominating. And this is one of the biggest issues with the chytrid fungus. It's causing what's called biotic homogenization. So all the chytrid areas are becoming increasingly, you could call it, homogenized. They all look the same, and the overall diversity is lost. So exactly as we expected, but we would probably also have expected to see a lower richness, but that's simply not the case in our data. So is eDNA really just the new golden standard for how you do biodiversity analysis? And the short answer is no. It is really, really efficient at certain things, such as detecting rare species. There are several cases where the eDNA has been compared to field surveys, where they manage to find rare species that field surveys don't, even though it's experts within the given category, the given field of biodiversity. eDNA finds those really rare individuals. But it has a lot of issues. So when you work with metabarcoding, it only gives you presence-absence data. So when I have a sample, I can't tell if there's 200,000 or just a single individual of that given species that I found. And looking at biodiversity, this is really important information. Secondly, in the case of our study, working with environmental DNA, we don't know anything about the individual infection of the amphibians. We can just see that these frogs are in the environment, and so is the chytrid, or maybe it's not. But it's very possible that many of the amphibians living in the environment are not even infected or has issues with it. So we still really need traditional field surveys. There's also the question, has a detected organism actually been there? Because these small DNA fragments, they can move around. In case a bird ate a tadpole somewhere on the Caribbean coast, it flew up here and took a shit in the lake, and it's gonna look like that frog has been there, even though it was never even close. And the same goes for rainwater. It can pull DNA fragments into lakes. We have cases in our study with species being present in lakes, although it's a species that's never associated with lakes. So it has a lot of pitfalls, but it is a great tool. One of the biggest issues when working with eDNA in the tropics is that the reference data is limited. So all the DNA sequences that we are analyzing, we're comparing them to a database. And there's just not a lot of data for tropical regions. This map here shows it pretty well. This is from the NCBI, which is the biggest DNA database that you normally use. And it shows where samples of rosids, a group of plants, have been taken and registered in NCBI. And each little dot is a sample. And as you can see, Europe is pretty prevalent, well studied, although it's the place, well, one of the places with fewest of this given group of plants, while Brazil is the most diverse place in the world in terms of rosettes. So it makes it much more difficult for us to work with environmental DNA in the tropics. And these are just some numbers to give you an idea of how understudied the tropics is in terms of genetic analysis. We know that at least 62% of all vertebrates, so all animals with a spine, they dwell in the tropics, and yet less than 40% of the genetic data is from the tropics. And the same goes for vascular plants. 70% of all species are here in the tropics, and less than 40% of the data is from the tropics. And it's even worse when you look at fungi. The good news is that the place that we're at, Monteverde Institute, now has a min-iron nanopore sequencing instrument, which is very efficient at sequencing. You can even do it in the field. So you just have to go find a project and look for some rare species and help the world understand biodiversity better. And that's it. This is the one fungus to rule the frogs, made by AI. It's pretty cool. Yeah, feel free to ask any questions. I'd like to disclaim I'm not a herpetologist, so I might not know everything about amphibian biology, but I'll answer to the best of my ability. Yes?
Did you look at any of the other qualities of like such measurements when you took pond and river samples? It was probably out
Can you repeat louder?
It was probably out of your scope to look at this, but when you took water samples specifically from like the rivers and ponds, did you look at anything else within that water, or just specifically use the genetic sequencing to simply just look for that, or did you look for possibly like pH or other nutrients?
No. That would be interesting to look at, because things like pH are really important for the degradation rates of DNA molecules. So ideally, I would have looked at that, but I just didn't have the equipment. Yeah. Yes?
Does the fungus attack the tadpoles in water?
Yes, it does, but usually it is not virulent towards the tadpoles because their skin is very different. So it actually only infects the mouth of the tadpoles. And what happens then is that quite often, when that tadpoles goes through metamorphosis, it becomes an adult that is immune to the chytrid fungus. So actually some places have tried to make these amphibian nurseries where they expose tadpoles to the chytrid fungus and then release chytrid-resistant adult frogs to the nature. But it's very time-consuming, labor-consuming, and it's not 100% efficient to my knowledge.
By chance, have they found if a chytrid-resistant frog, are the spawn also chytrid-resistant, or do they have to go through the same process?
I think they have to go through the same, yeah, unfortunately. I think it's not a genetic immunization of the frog, it's the microbial community on the skin of the frogs. Frogs have super complex microbial diversity on their skin, similar to our intestines. And it has been shown that some bacteria actually produce antidote to the toxins from the chytrid. And that doesn't pass on to no, unfortunately not. But yeah, good question. Yes?
So you said metabarcoding for amphibians doesn't reveal anything about abundance, but using eDNA for chytrid can reveal
Exactly.
abundance?
Yeah, it's
How does it do that?
two different techniques.
Okay.
So for the chytrid, we used qPCR, which is specifically for a single organism. In our case, BD. And this process where you amplify the genetic material from the chytrid, as you amplify the genetic material, it emits light. And the stronger the light emission, the more chytrid is present in that sample. And you then compare that with a number of samples where you know exactly how much of chytrid is present in that sample. So you call it a standard curve. And based on that standard curve, you can determine the abundance of chytrid in a given sample. But it only works for specific organisms. If I had done that with amphibians, it would only show me how much amphibian DNA is present in that sample. And that doesn't really tell very much because maybe it was full of cane toads and they're, it's a big toad. So it's going to shed a lot of DNA. And on the contrary, there might have been a lot of other frogs in it, but they were really tiny and it's not going to show in the method. Some people have suggested that you can use it to assess relative abundance. So like you can rank, okay, this is the most common and this is the least common. But again, it's an issue if it's two different sized animal, which is often the case with amphibians. Yeah.
So is it only really like microbes that it's suitable for?
Yeah, it's mostly microbes it's used for, yeah. Some people do it for salmons too, but again, there's a lot of uncertainty about the number that you get. Yeah, mainly microbes. Yes?
How were the soil samples collected? Like surface or?
Yes, strictly surface, like maybe half a centimeter down. And we would then freeze-dried them and powderized them and took DNA extracts from that. Yeah, so it'd be interesting to see what's deeper in the soil, but yeah, we didn't look at that. Anything else?
How will, based on what you did, does something comes to your mind that like do a second study or something different based on your data, or like any idea they have come after you get your results?
Yes, a lot. One thing I would really love to do is to combine my data with acoustic monitoring. So set out a speaker which monitors the sound, because most, at least the frogs, are very vocal. So that could give some kind of abundance estimate of how much is actually present of a given species. But that is also, they have different breeding seasons, they call at different times, so it would be much more difficult. It would also be cool to test some solutions to this. Last year, some Australian scientists created these frog saunas, kind of. It's like a black brick in a glass house with little holes in it. And like I said, some amphibians, they seek out warm areas when they get sick. And these black bricks get super warm and it's quite dry in there. And they climb in there and they clear off the disease. And it would be cool to set those out in some of the environments that we now know has a lot of the chytrid present and see if it actually has an effect. That's another thing. And also, I was initially planning on climbing the trees to collect samples from the bromeliads in the top, but climbing a tree is very time consuming, right? And we prioritized getting more locations over bromeliads in the trees. It would be cool to look at that. Yes, thank you very much for showing up.
Gracias, Tobias. Ahí Gabriel, acuérdense por ahí, tenemos café y té.
Very nice, man. Nice to see you.
I'm glad you liked it.